Hello, everyone, and welcome to this Labostix broadcast. In this, as part of this series, the broadcast edition. This is a Labostix release and a Labostix broadcast in around the Communicade, a chartered edition and evidence-based perspective, and another Storm Labostix production. In this overview, in around this broadcast edition, there'll be some overview, an overview, or some overview in around looking at some collaborative works again, works that was sort of originally presented at within the remit of the a biomedical science congress in 2015 in the United Kingdom, and this work was an overview in around nephrotoxic nephritis, glomerulonephritis, animal model versus human disease, and wherein this works was published in a journal in the Journal of the British Journal of Biomedical Sciences, and what was published in the British Journal of Biomedical Sciences as a review in around the particular subject in around nephrotoxic nephritis, and the Congress, the Biomedical Science Congress, offered an opportunity to present the work sort of within the remit of within the remit around sort of the sciences, where the remit was in around the sciences around various laboratory and pathology disciplines and specialties. So here goes. So the term glomerulonephritis encompasses a range of immune-mediated disorders that cause inflammation with the glomerulus and other compartments of the kidney. The mechanisms of interaction between these cells and the mediators of coordinated response to inflammation have been sort of elucidated and within literature. Despite these sort of pathophysiological advances, treatments for glomerulonephritis remain non-specific, hazardous, and only partly successful. Glomerulonephritis, or GN, therefore remains a common cause of CKD globally. Animal model approaches offer effective and safer treatments retrospectively and prospectively. Evidence of compartment deposition with glomeruli is a main feature of main forms of GN, and the role of complement is implied by several observations in human disease. Indirect evidence obtained from human studies has been supported by findings in animal models. The mechanisms by which macrophages mediate renal injury remains to be confirmed, but it is likely to include, or has likely to include, or has been likely to include a promotion of inflammation through release of pro-inflammatory cytokines such as Tumour Necrosis Factor, or TNF, recruitment of macrophages and other leucocytes through release of chemotactic molecules such as Monocyte Chemotactic Protein-1, or MCP1, cell proliferation by release of growth factors such as Macrophage-Colony-Stimulating Factor, MCSF, and cell death through the release of reactive oxygen species. In around this works in around this works some overview in around some bases in around the aims. So research with animal models have shown the sort of a crucial interaction between inflammatory cells and cells intrinsic to the kidney or kidneys that are fundamental and unique to the pathogenesis of GN. The objective of this works was to provide insight on sort of appropriate or suitable model in and around nephrotoxic nephritis and nephrotoxic nephritis and GN, and relate this or regard this in context of experimental validity in humans. So in around sort of the epidemiology around GN. Many cases of GN result in mild asymptomatic illness that is not recognized by the patient and remains and by patients or by patients and remains and can remain undiagnosed. The incidence and prevalence of such mild episodes of GN are unknown, but could be substantial. In the United Kingdom, the most common recorded one of the most recorded renal diagnoses is GN by biopsy, where there's biopsy proven and non-biopsy proven, um, and for all ages, the prevalence rate in male exceeds that in female, peaking in the age group between 75 to 79 years of age, and in around where evidence indicates around 2,918 per million population in males and for females, and for females the peak is in around GN in the age. It has been evidence in as the age group between 65 and 69 years of age at 1,000, and where it's evidenced as 1,460 per million population. Population-based screening studies or investigations have shown that evidence of renal damage by proteinuria, hematuria, low calculated glomerular filtration rate (GFR), or a combination of these features is present in around 16% of adults in Australia, and a similar proportion in the United States. And accurate classification requires a histological diagnosis, and consequently, the reported prevalence may or might vary according to local indications for renal biopsy. A study has informed renal biopsies done in the Australian state of the Australian state of Victoria during 1995 and 1997 had found that 21 people per 100,000 population per year underwent renal biopsy, yielding a yearly incidence of biopsy-proven GN of around 12.3 per 100,000. The most common diagnosed types or forms of GN in adults were IgA nephropathy, focal and segmental glomerulosclerosis, and vasculitis. Those most commonly diagnosed in children or young people were minimal change disease, focal and segmental glomerulosclerosis, lupus nephritis, and IgA nephropathy. In around sort of the pathogenesis then. The pathogenesis of GN is complex. Intricacy arises from several factors such as autoimmunity, cancer, and structural abnormalities within the kidney. There is a variability and susceptibility to GN, which is likely to have a genetic basis. That are also complex interactions between soluble factors, including antibodies, complement, chemokines, cytokines, and growth factors, and cells, including leucocytes and intrinsic kidney cells that mediate the sort of glomerular inflammation. The contribution of non-specific factors such as hypertension, and proteinuria promote continuing kidney damage despite resolution of initial insult. a constant feature of GN in the presence of inflammatory leukocytes within both the glomerular and the institutional compartments of the kidney. A constant feature of a GN is the presence of inflammatory leukocytes, and wherein this is evident within both the glomerular and the institutional compartments of the kidneys. Macrophages and T lymphocytes are perhaps the most dominant cell types present, and the intensity of cellular accumulation, particularly with the institution rather than the glomerulus, has repeatedly been found to correlate with the clinical and pathological severity of disease in animal models and in human beings. With the kidney T cells and macrophages undergo activation, within kidney, and the T cells and macrophages undergo activation, which is triggered by interactions between leukocytes and intrinsic renal cells, by interactions between cells matrix and cells matrix and cytokines and other soluble factors present within the kidney, present within the inflammatory environment. Type 3 hypersensitivity occurs, and where type 3 hypersensitivity occurs, antigens and antibodies are present in roughly equal amounts, causing extensive cross-linking. It is characterized by soluble antigens that are not bound to cell surface in the case of type 2 hypersensitivity, and when these antigens bind antibodies, large immune complexes from that cannot be cleared, the reaction can take hours, days, or even weeks to develop. Type 3 hypersensitivity represents the progressive forms of GN. In around sort of the animal models around GN and in context of NTN as well. An animal model is used during research and investigation of human diseases. The animal selected will usually an animal is usually selected the animal selected usually meets a determined taxonomic equivalency to humans, so as to react to disease or its treatment in the way that resembles human physiology as required. Research relating to nephrotoxic nephritis, or NTN, and glomerulonephritis can be traced back to the 1960s and the 1970s, when investigations reported observations on the role of complement in nephrotoxic nephritis, produced in rat laboratory models, but also by rat and rabbit and duck in around nephrotoxic sera, or NTS, in around these particular laboratory models. In contrast to commonly used animal models of immune complex mediated NTN-GN, BALB-C murine or mice models have been highly reproducible and only require short-term injections of lipopolysaccharides, and this BALB-C NTN murine BALB NTN murine mouse models still holds experimental validity in the study of GN in humans. In addition, the BALB murine mice models can be helpful in the investigation of GN autoimmune phenomena. In addition, duplicating features of human GN have produced insights into the pathogenesis. The reproducibility and uniformity of such animal models has permitted a sort of a step-by-step dissection of many of the mechanisms involved. In around the role of chemokines as well. It is now to be appreciated that when glomeruli are damaged by inflammation, growth factors are filtered into urine where into the urine where they activate renal tubular cells, thereby causing them to secrete chemokines and cytokines. Intriguingly, there has been a team that generated CXCR3, a particular chemokine CXCR3 deficient murine mice models to investigate the functional role of CXCR3 in the kidney under pathological conditions. NTN was induced to analyze the molecular basis of T cell recruitment in GM by CXCR3 and its three ligands IP10, CXCR10, MIG or MIG, CXCR9, and ITAC or CXCL11, and to identify potentially novel targets for therapeutic interventions. So this research had evidence that chemokine receptor CXCR3 mediates renal T cell recruitment and subsequent tissue injury in NTN. And the investigation provided a rationale for considering CXCR3 blockade in TH1 type TH1 type mediated cells and in renal inflammation as prospective therapeutic options for patients diagnosed with GN and GN subsets. In around sort of the treatment aspects, regular clinical follow-up in line to sort of the clinical care pathway in patients where there's evidence of proteinuria exceeding 1 gram per day, 1 gram daily has been evidence in literature as being advantageous. Findings have suggested that more complete blockade of the renin-angiotensin system by use of both angiotensin-converting enzyme inhibitors and angiotensin receptor antagonists is superior to either agent alone for prevention of progressive chronic kidney disease amongst patients with proteinuria. Treatment of other symptomologies has slow progression in experimental models of GM, but available trial data are insufficient to prompt sort of the recommendations specifically for renal protection, where in around where data has not been fully complete. Currently used immunosuppression therapies, for example, for human GN are not always uniformly effective and are frequently associated with serious side effects. Corticosteroids are effective in several types of GN owing to ability to inhibit pro-inflammatory effects of cytokines known to promote glomerular inflammation, including interleukin 1 beta or IL-1 beta and TNF, Tumour Necrosis Factor alpha. So in around the sort of conclusions around this works, the objective of this works, or this article even, was to provide insight on sort of the suitable appropriate model in around nephrotoxic nephritis and nephrotoxic nephritis and glomerulonephrosis, glomerulonephritis, glomerulonephritis, and relate sort of the experimental validity in humans. Animal with experimental NTN-GN are homogeneous, meaning that they're the same or quite similar when animal models are used in context of laboratory investigation, whereas patients and human GN are heterogeneous, whereas there are differentiations around GN in context of human GN in context of GN and diagnoses as well. Experimental disease is initiated at a time determined by investigators, and runs on a predictable basis, thereby permitting intervention at defined stages, including before induction, during disease establishment, or later. Patients typically have established disease before treatment can be prescribed or administered. Renal damage tends to be progressive in the longer term, despite elimination of the original source of injury. Differences between experimental and human GN complicate translation of experimental therapies into practice. Stem cell based therapies should be explored. These could include genetic modification to produce protective molecules within the kidney, or to promote regeneration of damaged intrinsic kidney cells. There is a need for more up-to-date research on this topic, and further research or work is required to translate animal model research to better comprehend GN disease in humans. The complexity of GN research makes findings a challenge to replicate or duplicate more consistently. However, finally, there are more continuing evidence-based sort of assessments in around the immune and non-immune therapies, or direct or rational care for rational sort of pathways or care for patients or recipients. This works is available through number one, through the Renal Patient Support Group, the RPSG, and also available as a publication in the British Journal of Biomedical Sciences, BJBS, the BJBS, British Journal of Biomedical Sciences. Alongside with this broadcast edition, there will be some resources in the descriptives as part of this overview. So listeners are welcome to check out that for further information around this works. And once again, this works in around sort of looking at nephrotoxic nephritis and glomerulonephritis, animal model versus human disease, as an overview or a review, and originally was presented at a biomedical science congress in 2015, in the United Kingdom. So once again, thank you for tuning in. This has been a Labostix broadcast, part of the broadcast edition, a Storm Labostix production, a Labostix release in around the Communicade, a chartered edition, and an evidence-based perspective. Thank you for tuning in.